The year is 1928, a time when the world is still recovering from the Great War and grappling with the challenges of modern medicine. In the bustling city of London, amidst the foggy streets and the hum of industrial progress, a quiet revolution is about to begin. Inside a cluttered London laboratory, where the air is thick with the smell of chemicals and the hum of scientific curiosity, Dr. Alexander Fleming, a Scottish bacteriologist with a sharp mind and an unyielding dedication to his craft, is hard at work. Fleming, renowned for his meticulous research and keen observation skills, is deeply engrossed in studying Staphylococcus bacteria, a common yet stubborn pathogen. His workspace, though chaotic, is a reflection of his relentless pursuit of knowledge. Petri dishes, test tubes, and notes are scattered across the room, each dish hosting a unique colony of bacteria, carefully nurtured and observed for days, sometimes weeks. Little does Fleming know, amidst the apparent disorder of his lab, fate is quietly orchestrating a groundbreaking moment, a discovery that lies hidden in plain sight, waiting to be unveiled by his sharp eyes and inquisitive mind. The summer of 1928 is unusually warm, and Fleming's lab, like many others of the time, lacks modern conveniences like air conditioning. The warm, humid air creates an ideal environment for microorganisms to thrive, as dust particles and spores float freely, settling wherever they please. Among these uninvited guests is a tiny spore of a rare mold, drifting unnoticed into Fleming's lab and landing on one of his petri dishes. This seemingly insignificant event sets the stage for a revolutionary discovery, one that will not only transform the field of medicine, but also save countless lives in the years to come. Upon returning from a holiday, Fleming notices something peculiar in one of his discarded petri dishes. It's a moment that would change the course of medical history forever. A blue-green mold has taken up residence, contaminating his experiment. But this contamination is no ordinary mishap, it's a clue waiting to be unraveled. While most researchers would discard a contaminated sample, Fleming's curiosity is piqued. He decides to investigate further, driven by an instinct that this anomaly might hold deeper significance. He observes the dish more closely, noticing a clear halo surrounding the mold. This halo isn't just an oddity, it's a sign that something extraordinary is happening. The Staphylococcus bacteria he was cultivating have vanished in this zone, inhibited by the mysterious mold. It's as if the mold is waging a silent war against the bacteria. Fleming's sharp mind recognizes the significance of this chance observation. He begins to wonder, what if this mold could be harnessed to combat infections? Could this mold hold the key to fighting bacterial infections? Could it be the breakthrough the world has been waiting for? He identifies the mold as Penicillium notatum, a species previously overlooked for its potential, and dedicates himself to understanding its properties, determined to unlock its secrets. He conducts further experiments, isolating the active substance responsible for killing the bacteria. This substance, he discovers, is both potent and selective in its action. He names this substance penicillin, marking the birth of the world's first antibiotic. This discovery would go on to save millions of lives, a testament to the power of curiosity and serendipity. Fleming publishes his groundbreaking findings in 1929, but the scientific community initially greets his discovery with skepticism. The process of extracting and purifying penicillin in sufficient quantities proves to be a daunting task. Fleming lacks the resources and expertise to fully develop penicillin into a usable drug, hindering its immediate application. Despite the challenges, Fleming remains convinced of penicillin's potential. He continues his research, tirelessly experimenting with different methods to produce and purify the drug. He even attempts to treat some of his patients with crude penicillin extracts, achieving mixed results. However, his efforts to mass-produce penicillin are met with limited success. The world would have to wait for over a decade for penicillin to realize its full, life-saving potential. Section 4. The collaborative spirit of discovery. Florey and Chain join the race. The year is 1939, a time of global uncertainty and rapid change. The world is on the brink of a devastating conflict, and science is about to play a pivotal role in shaping the future. World War II has begun, and the need for effective treatments for battlefield infections is, well, more pressing than ever. Soldiers face not only the horrors of war, but also the silent threat of infections that claim lives as swiftly as bullets. The medical community is desperate for solutions. Across the Atlantic, at Oxford University, two scientists, Howard Florey and Ernst Chain, stumble upon Fleming's forgotten paper on penicillin. This paper, published a decade earlier, had largely been overlooked, but to Florey and Chain, it held immense promise. Intrigued by its potential, they embark on a mission to turn Fleming's discovery into a usable drug. It was no small task. Penicillin was notoriously unstable and difficult to work with, yet they were undeterred, driven by the hope of saving countless lives. Florey, a pathologist with a keen understanding of disease, and Chain, a biochemist with a talent for unraveling molecular mysteries, brought their unique expertise to the challenge. Together, they formed a formidable team, united by a shared vision. They meticulously develop a complex process to purify and mass-produce penicillin. This involved countless experiments, trial and error, and an unwavering commitment to their goal. Their work was groundbreaking, laying the foundation for modern antibiotics. Their efforts are fueled by a sense of urgency, knowing that each passing day on the battlefield means countless lives lost to infection. The stakes were high, but so was their determination. Florey and Chain's work would soon change the course of medicine and history forever. Section 5. Penicillin's Baptism by Fire, a beacon of hope on the battlefields of World War II. In 1941, Florey and Chain conduct their first human trials of penicillin, achieving remarkable results. The drug proves to be highly effective against a wide range of bacterial infections, including pneumonia, meningitis, and sepsis. News of penicillin's miraculous effects spreads like wildfire, and soon the demand for the drug skyrockets. The mass production of penicillin becomes a top priority for the Allied forces. Pharmaceutical companies in the United States and Britain join forces, working tirelessly to meet the overwhelming demand. Penicillin is hailed as a wonder drug, saving countless lives on the battlefields and profoundly impacting the outcome of World War II. Section 6. A legacy of life-saving innovation. Penicillin's enduring impact on modern medicine. Penicillin's impact extends far beyond the battlefields of World War II. Its discovery ushered in a new era of medicine, the age of antibiotics. Bacterial infections that were once deadly became treatable, saving millions of lives worldwide. The development of penicillin paved the way for the discovery of numerous other antibiotics, revolutionizing the fight against infectious diseases. Today, penicillin and its derivatives continue to be widely used to treat a wide range of bacterial infections. From ear infections in children to life-threatening sepsis in hospitalized patients, penicillin remains an indispensable tool in the arsenal of modern medicine. Section 7. The enduring lesson of serendipity, a testament to the power of observation. The story of penicillin is a testament to the power of observation, scientific curiosity, and the collaborative spirit of discovery. Fleming's keen eye and willingness to explore the unexpected led to a chance encounter that changed the course of history. Florey and Chain's dedication and tireless efforts transformed a laboratory curiosity into a life-saving drug. The discovery of penicillin reminds us that breakthroughs can occur when we least expect them. It underscores the importance of fostering a culture of scientific inquiry, where serendipity and meticulous observation go hand in hand, leading to innovations that benefit all humankind.